Next, we'll be joined by Munmun Bhasin from the Indian Institute of Science in Bangalore. She has a pre-recorded talk, but will be taking questions afterwards via Zoom.
Hello everyone. Today I would be talking about structure and stability predictions from saturation mutagenesis. To begin with, I'll walk you through the deep mutational scanning framework, which begins with the target selection. This is followed by the construction of site saturation mutagenesis library, in which every residue is mutated to all the other 19 amino acids. This is followed by a selective enrichment, which could be a phenotypic readout, or a protein display assay, in which the binding and expression can be checked on the yeast cell surface. This is then followed by the high throughput sequencing of the variants, which is before and after the selection. So this basically gives us the idea of the fitness estimate for each of these variants, and this mutational landscape can be used to identify the substitution preferences at different positions. Taking this idea ahead, we utilize the information from the mutation sensitivity data of the published datasets for the prediction of the active site, buried and exposed non-active site residues. We combine the scores of the mutational effect with the accessibility prediction based on the sequence. Thus, we could identify the active site and the buried residues. So here we could see that the active site residues, they gave quite good results with respect to their specificity and accuracy, and they could be segregated from these buried positions. Also, we could clearly discriminate between these exposed non-active site positions. Thus, we concluded that the high sensitivity values in the exposed non-active site prediction results suggest that this method is capable of recognizing the mutationally tolerant positions from those which are mutationally sensitive. Now, this prediction of the buried residues is solely done from the mutational sensitivity data also, but it can be enhanced by incorporating the predicted sequence-based accessibility values to distinguish between these buried and active site residues. But on the other hand, these predictions seem to be inaccurate for the proteins which have limited mutational sensitivity data available. To validate these results further, we deep sequenced the CCDB library and we performed the mean fluorescent intensity calculation for these CCDB mutants. So the CCDB is basically a toxin component of the CCTAB toxin antitoxin system. So what we did was we did the yeast surface expression and the sorting of the CCDB single site saturation mutagenesis library. We checked the binding with gyrase A14 at 2 different concentrations and also checked its expression. This was further subjected to deep sequencing, and then the MFI reconstruction was done from the deep sequencing data. So what we can see here is for the exposed non-active site residue, these mutations did not affect either the degree of the surface expression or the binding to gyrase A14. On the other hand, when we look at the buried residues, we see that there is a high mutational sensitivity which is there for both expression as well as for binding. On the other hand, when we look at the active site residues, we see that they show mutation sensitivity only in the case with respect to the gyrase binding, whereas the expression it seemed to be unaffected by the mutations in the active site residues. So the buried site residues with very high mutation sensitivity, both in the case of the expression and binding, could be segregated from the active site residues that show mutation sensitivity only with respect to the gyrase binding. Now, this information about mutational sensitivity of expression and binding can be used to differentiate between exposed, buried, and active site residues. We further used a new parameter, which was MRMFI, that is the mean residue MFI, that was defined, that was basically taken as the mean of the MFI values for the mutants at a certain position. This was further made more sensitive by calculating the MRMFI charge, which was taken only for the mutants with the opposite charges. So thus we can see here that the prediction results for the MRMFI charged showed good results both for the case of the buried as well as the active site residues. We also tried this for another unrelated protein, which was the RBD of the SARS-CoV-2 from a previous report of the deep mutational scanning data. And here we also see that as we got good results with respect to the specificity, sensitivity, and accuracy for CCDB, similarly we got good results for the RBD as well. Thus, this average MFI-seq values of the charged can be used as a good predictor to discriminate between buried and active site residues. And it can be done for another unrelated protein, which is the receptor binding domain of SARS-CoV-2. To conclude, the analysis of available deep mutational scanning datasets revealed patterns of mutational sensitivity and substitution preferences at buried and exposed positions. MFI-seq was used to generate the mutational landscape of both expression and binding of the mutant library, and this was used to discriminate between the berate, exposed non-active site and exposed active site residues for CCTB, as well as for RBD of the spike protein of SARS-CoV-2. Finally, I would like to thank my PI, Professor Raghavan Vardarajan, and all the other lab members who are involved with this work. I would like to thank the funding agencies, CSIR, Department of Biotechnology India, Ministry of Education India, and IISc. Thank you so much.
Thank you again, Munmun. We have now time for a couple of questions.
Hi, thank you. It was interesting to see aggregating the score of charged residues, and I wonder if you saw in your sort of analysis of patterns how, if you could see how often it mattered only that it was charged versus it needed to be positive or negative charged?
Okay, so what we did was that with respect to the analysis that we had done, so in the cases when we had the calculations which were done for the charged residues, then we took the analysis for the opposite charges that are there for the analysis, and definitely we found that the sensitivity of the results were more with respect to these positive and negative charged residues. So definitely they gave quite a good impact with respect to the mean residue MFI calculations, and definitely if somebody wants to increase the sensitivity of their analysis, then considering these charge substitutions could be a good alternate for their analysis. Yeah.
Okay, thank you again.
Yeah, thank you so much. And I'm really thankful to the organizers for hosting this hybrid event. And it's great to join everyone from India here. Due to the issues in the visa, I could not come there. But yes, it was a great event. And thank you so much. I would like to discuss this further over my poster. So please stop by on Thursday. And thank you so much.
Yeah, we're looking forward to seeing you on Thursday.